Students, a very good afternoon to you. Happy new month and happy weekend. So this afternoon we are going to be looking at MLS 533 enzymology, talking about mechanics of enzyme action and kinetics, mechanics of enzyme action and kinetics. So before we go on, we'll first of all look at what an enzyme is. We should know what an enzyme is before we proceed. So enzymes can be defined as biological polymers that catalyze biochemical reactions. Take note, they are biological polymers that catalyze biochemical reactions. So there's hardly any reaction you see that takes place in the body that is not being mediated by enzymes. You know, almost everything. When you talk about digestion, for example, in the body, you must mention some enzymes. There are enzymes that are responsible for digestion, enzymes responsible for every other thing that happens in the body, that happens concerning biochemical reactions. So these enzymes in the body, you know, they function, they are in form of protein. They are in form of protein. So there's a saying that all enzymes are what? Are proteins. They are proteinous in nature. But not all proteins are what? Are enzymes. Okay. So these enzymes, they have the functions that they carry out in the body. They have functions they carry out in the body. Number one is the enzymes help in signal transduction. They help in signal transduction. The most common enzymes that are used in this process include protein kinase that catalyzes the phosphorylation of proteins. Take note, I mentioned protein kinase. They are enzymes, protein kinases. They catalyze the phosphorylation of proteins. Two, another function of enzymes, they break down large molecules into smaller substances that can be easily absorbed by the body. They break down, just like galactose, for example, it's not absorbed in the body as galactose, it is being broken down. So enzymes are responsible for the breakdown of large molecules into smaller substances that can be easily absorbed in the body. Three, they help in generating energy in the body. They generate this energy in form of ATP. So they help in generating energy in the body. ATP synthase is the enzyme involved in the synthesis of energy. ATP synthase, that's another type of enzyme that we've mentioned now. You know, when you hear enzyme, most time they end with their suffix is usually ASE, they end with ASE. So ATP synthase is the enzyme involved in the synthesis of energy. Four, enzymes are responsible for the movement of ions across the plasma membrane. So across the plasma membrane, ions have to move. And for there to be movement of ions across the membrane, it has to be mediated by what? Enzymes. Five, enzymes perform a number of biochemical reactions, including oxidation, reduction, hydrolysis, etc., to eliminate the non-nutrient substances from the body. Six, they function to reorganize the internal structure of the cell to regulate cellular activity. So you see that enzymes in the body have a lot of roles. They have so many roles they play in the body. So I want us to just quickly look at enzymes briefly before we now talk about their mechanism of action. So because enzymes are proteins, of course, they have some factors that affect them. But before we get to that place, we'll look at what we call an active site. An active site. Enzymatic catalysis depends upon the activity of amino acid side chains assembled in the active center. Enzymes bind the substrate into a region of the active site in an intermediate conformation. Now, what do we mean? Enzymes have what we call an active site, a place where they can bind. A place where they can bind. If that active site is not there, reaction will not take place. Just like, for example, somebody wants to enter into a house. Your key has a hole where it has to fit in. So if it's a round key, it should be if it's a round peg, in a round hole. If it's a square peg, it should be in a square hole. So if that active site is not present, catalytic reaction cannot take place by enzymes. Now, often the active site is a cleft It's a cleft or a pocket. It's a cleft or a pocket produced by the amino acids which take part in catalysis and substrate binding. Amino acids forming an enzyme's active site is not contiguous to the other along the sequence of primary amino acid. The active site amino acids are assembled in the cluster in the right conformation of the three-dimensional folding of the primary amino acid sequence. You know, of course, amino acids, you know, they are in sequential form of sequence, so in form of a spiral pattern. So in that spiral pattern, they have bonds that bind them. So these active sites of amino acids now are assembled to the cluster in the right conformation of the three-dimensional folding of the primary amino acid. So we have talked about the active sites of an enzyme. When we say there's a place where it has to bind, you understand, if that place is not there, it cannot bind, and catalytic reaction cannot take place. Now we have factors that affect enzyme. We have temperature and pH. Of course, we know that temperature, when the temperature is high, it can denature an enzyme. So enzymes require an optimum temperature and pH for their action. The temperature or pH at which a compound shows its maximum activity is called optimum temperature or optimum pH. So at that pH, they show their maximum activity. Anything outside that is something else. So as a temperature or pH more than optimum may alter the molecular structure of the enzymes. Generally, an optimum pH for enzymes is considered to be ranging between 5 and 7. That is the optimum pH, slightly acidic. So between 5 and 7, that is the optimum pH for enzyme reaction. Okay, so move on then. The greatest number of for that of the temperature, you know, human enzymes, their normal temperature is between 35 degrees to 40 degrees Celsius. 35 degrees to 40 degrees Celsius. And we know normal human being body temperature. Our normal body temperature is how many? It's 37 degrees. So and that is the normal temperature at which it can function properly. So anything that increase beyond the optimal temperature will denature the enzyme. So denaturation, and enzymes lose their 3D shape. Their 3D, that is the three-dimensional shape, when the temperature is outside the optimum temperature. Cold can decrease. Of course, we know it can. Decrease in temperature also affects enzyme activity. Enzyme will not be able to function properly. So another thing apart from temperature and pH that can affect the activity of an enzyme is concentration and type of substrates. Now under it, enzymes have a saturation point. That is, once all the enzymes added are occupied by the substrate molecule, its activity will cease. When the reaction begins, the velocity of enzyme action keeps on increasing on further addition of substrates. However, at a saturation point where substrate molecules are more in number than the free enzyme, the velocity remains the same. So the type of substrate also is another factor. Type of substrate that has to bind, in order for enzyme reaction to take place matters. So if it's not the right substrate, of course, the binding will not be effective. Yes, the concentration. Once all the areas where enzyme is supposed to bind are all occupied, any other free enzyme will not have a place to bind. So that's what we mean by concentration and type of substrate. They also have salt concentration as one of the factors that affects enzyme activity. Changes in salinity. When you addition or removal of cations and anions can affect enzyme activity. We say it disrupts their 3D shape. It disrupts attractions between charged ion particles, charged amino acids. It affects their three-dimensional structure. The salt concentration, whether increasing or decreasing beyond the optimum concentration, denatures protein. Enzymes are intolerant of extreme salinity. When the salinity is extreme, enzymes cannot tolerate. So we have mentioned the functions of enzyme. We've talked about some factors that can affect enzyme activity, and we have also talked about the active site and talked about what enzymes are. So in our next class, we'll be looking at the mechanism of action of enzymes. See you in my next class. Bye.